Yeah, okay. Welcome to our monthly webinar series, and we hope you stay safe and healthy during this tough time of COVID-19. So we'd like to invite Ashok from NOAA today, and thanks Ashok for joining us today. And I'm going to introduce uh, him a bit. So Ashok Deshpande is a research chemist at NOAA Fisheries, Northeast Fisheries Science Center, James Howard Marine Science Laboratory in Sandy Hook, New Jersey. For about 30 years, he has been working on assessment of persistent chemical contaminants in the recreational and commercial fisheries resource species. He has examined the potential uses of intrinsic and naturally embedded trace chemicals in the assessment of habitat ecology, migratory behaviors, and predator-prey interactions. His current research is focused on the applications of chemistry principles in addressing the baseline fisheries topics, including the chemical characterization of microplastic polymers by using a novel techniques of pyrolysis, GCMS. He has successfully analyzed a variety of plastic samples from the littoral and aquatic environments and in seabirds as well. And uh, as always, we have the slide that is open for questions, and you can uh, ask questions anytime during his presentation or at the end of his presentation. And we'll go through the most liked question. So if you see the question on Slido, you can go to Slido, just click sli.do and hashtag is OPW, or you can find a link of the Slido below the screen of YouTube and just click it, and the keyword is OPW, and I pick up the most liked ones that, so you can see the like button next to the question, and if you click it, and I will pick up the most liked ones that you'd like to see answered. Okay, so Ashok, thanks for joining us today again, and you can share your screens now, and floor is yours.
Well, thank you, Dr. Ryota Nakajima, for inviting me to the Ocean Plastics webinar series. Appreciate the kind introduction. Greetings and a very happy new year to everyone. Hope you are all safe and healthy. A big thank to you all for attending today's webinar despite your busy schedules. It is a pleasure and an honor to share with you our research on the microplastic aspects. NOAA Sandy Hook Laboratory is located in Sandy Hook, New Jersey, which is across from water from New York City. There are several historical, military, and natural landmarks in the area. It is very difficult to imagine a life without plastics. Plastics are used in the household items. Plastics are used in construction industry. PVC appears to have about half the share of the plastics.
Hi, Ashok. It's
Yes.
in presentation mode. Can you change the? I can see your note.
Oh, you are?
Yeah.
Okay. Is it better?
Yeah, it's better. Thank you.
So plastics are used in the automobile industry. PVC appears to be increasing in the plastic production. Plastics are used in the toys, and plastics are used in the medical devices. The global footprint of plastics has increased exponentially over the last 50 years. When the production and the recycling are about the same, we have a good balance and there is negligible waste. But when the production exceeds recycling, we are off balance and waste is generated. This is an illustration. The red line includes plastic generated. The green line indicates plastics recovered and the yellow area shows the plastic trash. So as you can see, most of the plastics is trashed. There are about 270 million tons of plastics produced each year globally. About same amount of plastics is wasted. The coastal communities produce about 100 million tons per year, and out of which about 32 million tons are mismanaged. So about 8 million tons of plastics are input to the ocean every year. The great plastic tide appears to be encroaching our environment, and the scientists fear that we are entering a period of when the mermaids cry. The larger plastics are broken down into smaller plastics by UV light, wave action, wind, and the plastics which are smaller than 5 millimeters are called microplastics. Microplastics are also originated from the clothing of the synthetic fibers. The microplastics are originated from the noodles and also personal hygiene products. Microplastics are a cause for concern to the marine life because microplastic size overlaps with the preferred particle size ingested by the animals at the base of the marine food web. Detritus suspension and filter feeders can easily ingest them. It may lead to the uptake and trophic transfer of plastic themselves and any chemicals they may contain or have absorbed from the seawater. Many of these species are important to the fisheries or perform vital economic ecosystem functions. Microplastics can transport invasive species, including the harmful algal blooms and pathogens. Toxic additives, POPs, and monomers can be released in the environment when plastics are incinerated or sewage sludge contaminated with microplastics are used for energy production. It is important to note that not all plastic is created equal. Different plastics have different chemistries, they have different properties and applications, they have different fates and effects, and they have different management practices. The analytical tools available to chemists are infrared spectroscopy, Raman spectroscopy, differential scanning calorimetry, and pyrolysis GCMS. Infrared spectroscopy is based on vibrational excitation of covalently bonded atoms and groups. And we get a nice spectrum which is characteristic to plastic polymer. In the Raman spectroscopy, a monochromatic laser beam is irradiated on the sample. When when the laser hits the molecules, it is either absorbed, reflected, transmitted, or scattered. The scattering, the Rayleigh scattering, uh, the energy of the incident photon and scattered photon is about the same, while 1 in 10 million photons is scattered at frequencies which is different from the incident photon. Is called Stokes-Raman scattering. So this is used for determining the structure of polymer. In differential scanning calorimetry is based on thermal behavior of polymers. The instrument is based on a thermal cell which has a sensor that registers temperature between sample filled pan and the reference pan which contains only air. As we increase the temperature, the polymer undergoes various phase transitions, and depending upon if it is endothermic or exothermic, different amount of heat is needed to keep the two pans at the same temperature. So this is characteristic to a given polymer. The pyrolysis GCMS, that article that inspired me, was authored by L.K. Fries. The pyrolysis GCMS method includes a small plastic piece which is cut and placed in a quartz tube. The quartz tube is placed in a platinum filament of a pyroprobe. The pyroprobe is inserted in a GCMS interface. The filament is heated at high temperature like 750 degrees Celsius, and the degradation products are analyzed by GCMS. The sample is separated on a gas chromatographic column and analyzed by mass spectrometer to give a pyrogram which is indicative of the polymer. So these are the examples of various commonly used plastic polymers. The observations are that the pyrolysis GCMS pyrograms are different for different polymers. Similar to infrared and Raman spectroscopy, different pyrograms will allow differentiation between different plastic materials. Although more peaks may be detected in the pyrolysis GCMS pyrograms compared to infrared and Raman spectroscopy, the peak fingerprinting can be viewed as a qualitatively equivalent among the three techniques. So the question is, why do we bother with so many techniques? And what should I do? Which technique should I use? Should I play a guessing game and hope for the best result? Or should I find how to reach the target successfully? The spectroscopic methods, they actually work very well for many different types of samples. However, there can be some difficult samples that can pose challenges to the spectroscopic methods. These challenges can be related to the sample color, especially a dark color, or fluorescence, or surface properties, or presence of excessive amounts of additive chemicals. These interferences can deteriorate or smother the spectrum of the plastic polymer. The challenges can be instrument related or library database related. The high-end instruments have more features and they offer flexibility in data processing. The challenges can also be related to the skills of the instrument users. Compared to the novice, a skilled scientist will know exactly how to process the data and eliminate the interferences. Mass, Here are some of the benefits of pyrolysis GCMS. Mass spectrometry is a gold standard for the analytical chemistry data quality, and it provides the most accurate identification of chemical composition and structure. Many laboratories already have, and they routinely use various GCMS systems. There is no need to learn a new trade or find funds for the new instrument purchase. Addition of pyrolysis component to the GCMS is simple and cost efficient. Inserting a sample in the quartz tube is easy as threading a needle. Pyrolysis GCMS provides a two-tier confirmation approach, unique peak fingerprints of a given polymer, and mass spectrum of each individual peak. The two-tier confirmation approach provides higher confidence in the data quality. No sample preparation is required except for cutting a tiny piece for analysis. There is no effect of shape, size, density, texture, surface, color, brightness, opacity, weathering, etc. on the data quality. Information on chemical additives can be obtained in the same sample run. And the additional benefit includes the mass spectral library is huge. There are more than 267 compounds. So it is easy to detect the polymers, copolymers, and the additives in the samples. So we'll look at some applications of pyrolysis GCMS. We will look at the peak fingerprints. We'll look at the mass spectrum of each peak, and we will determine what polymer we have, if there is any copolymer, and if there are any additive chemicals. So this micropipette tip, which is commonly used in the laboratory, has a marker peak of polypropylene. When propylene molecule polymerizes, it forms polypropylene. This plastic piece has evenly spaced triplet peaks, which contains dienes, monoines, and saturated dialkanes. It is a signature of polyethylene. This is a zoom picture of the previous chromatogram, which shows the triplets that are comprised of double bonded, single bonded, and saturated hydrocarbons. Again, polyethylene molecules polymerize to form ethylene molecules polymerize to form polyethylene. The CD storage case has marker peaks of styrene, so we call it polystyrene. When styrene molecules polymerize, it forms polystyrene. The hair comb has a marker peak of polypropylene. It also has a marker peak of polystyrene. So it's the copolymer of polypropylene and polystyrene. This weed whacker trimmer line, which is very commonly used by many handymen in their yards, it has a eta caprolactam marker peak, which is indicative of nylon 6. Nylons are polyamides. The nylon 6,6 is prepared from polymerization of adipic acid and hexamethylene diamine, while nylon 6 is prepared by polymerization of caprolactam. Proteins are also polyamides. They are formed by the polymerization of different amino acids. This foam sample has a marker compound for polyurethane. Polyurethanes are formed by the polymerization of methylene diphenyl diisocyanate and ethylene glycol. They contain a unique urethane linkage. This beverage bottle has a signature of polyethylene terephthalate or PET. PET is formed by polymerization of terephthalic acid and ethylene glycol. PVC has very interesting pyrogram. It contains hydrochloric acid and some aromatic compounds. It is understandable how we get hydrochloric acid because it contains chlorine molecules, but it is not understandable why it gets aromatic compounds. This reminds me of a Aristotle quote: "The whole is greater than the sum of the parts." So we have the parts, but we cannot explain the whole. We can try to explain this now. Pyrolysis is a very high energy environment, and there is plenty of energy available for making and breaking of the bonds. So the hydrochloric acid is removed from the polymer chain. The bonds rearrange, cyclize, and the side chains are removed to form a stable benzene molecule. So that is the story of PVC pyrogram. The rubber band has limonene and isoprene as their marker compounds. So we call it natural rubber. The natural rubber is formed by the polymerization of isoprene. This water-soluble packing peanut has signature of cellulose. Cellulose is formed by polymerization of beta-D glucopyranodes. The last example probably has styrene and other aromatic compounds and also has methyl methacrylate. So we call it styrene methyl methacrylate. We will switch gears to look at some of the collaborative studies on microplastics. We work with academia, government, NGO, international scientists. We also work with National Science Foundation Fellows. We work with NOVA Educational Partnership Program with minority serving institutions, scholars, and NOVA hauling scholars. It literally takes a village to undertake studies of this magnitude. The plastics from New Jersey were given to us by Dr. Swarna Muthukrishnan and Cindy Zip. This plastic tip contains signature of polyethylene. This weathered plastic bottle also has signature of polyethylene. We were very surprised to see that the fist of fury and of Mother Nature banged up so much, but it did not make any difference in our data quality by using pyrolysis GCMS. This ballpoint pen contained the signature of polypropylene. So we went to work with Dr. Frank Hernandez of University of Southern Mississippi. He collected samples for us from sargassum mats in Gulf of Mexico. Sargassum mats are a proxy for the presence and movement of microplastics and marine debris. The first sample contained signature of polypropylene. The second sample contained signature of polypropylene. Dr. Steve Carr from the Los Angeles sanitation districts, he sent samples of some textile blends. The first sample had signature of polyethylene and also had a signature of cotton. So we called it terrycot or blend of polyester and cotton. The second sample contained signature of polypropylene and it also had signature of PET. So we call it blend of PET and PP. The third sample only contained the signatures of polyethylene terephthalate. So we call it PET. We received samples of from plastic samples from Kim Cloker from USGS. The samples were collected in the remote shorelines of northern Gulf of Alaska. This black foam contained styrene signature called polystyrene. This blue tape was interesting. It had a signature of polyethylene, but in addition to that, we found the isomers of tricresyl phosphates, which are used as plasticizers and flame retardants. These compounds also, this sample also contains some kind of steroids. This big lighter found on Alaska beach, it helped us add one more compound to our library list. It's called polyoxymethylene polymer or POM. Captain Charles Moore discovered a massive area of plastic soup floating in the Pacific Ocean in 1997 that sparked what we call now the Great Plastic Awakening. So Captain Moore and Gwen Latin provided us some samples of microplastics from the Pacific garbage patch. The first sample has signature of polypropylene. Second sample has signature of polyethylene. This sample is called ethylene vinyl acetate copolymer because it contains acetic acid and also has the signature of polyethylene. There are some additional peaks which we plan to analyze when we are able to go back to the laboratory. Then we go to the streams in the California. Win Cowger is the National Science Foundation doctoral fellow, and his advisor is Dr. Andrew Gray. The first sample has signature of polyethylene. This sample has a signature of polypropylene. I'm showing only few samples from each project because in each project we analyze between 30 to 140 samples. So in the interest of time, I'm not able to show all data. This ball size foam was polystyrene. This was an interesting sample. It looked like a glitter. It had signature of methyl methacrylate, butene, and styrene. So we call it methyl methacrylate butadiene styrene copolymer. Dr. Jennifer Lynch and Kayla Brignac provided us samples of plastics from the Hawaiian beaches. This sample contained styrene, acrylonitrile, and butadiene. So we call it ABS or acrylonitrile butadiene styrene. This sample contained polypropylene and polystyrene. The sample from KUHI contained cyclopentanone. It is a marker compound for nylon 6,6. This sample from Hawaii was interesting. It showed signature of cellulose. In addition, it had a peak of cannabinol, and it also had a peak for Myrex or ketone. Next sample contained isoprene and limonene, and also a additive called phthalate. So we called it latex plus phthalate additive. We got some samples of seabirds from Dr. Chris Pham and Yasmina Maria Rodriguez Gonzalez of University of Azores. The samples were collected from 4 islands in Azores Islands: Pico, Sao Jorge, Fayal, and Flores. Seabirds are again indicators of the ocean health and disturbances. Oh, we work with Dr. Chelsea Rockman and Dr. Rebecca Sutton. They were working on plastic particles in the San Francisco Bay, and they wanted to find out if the samples they gave us were uh, they originated from the tire trades. So some tire talk. Main polymers used for the tires are natural rubber, styrene butadiene rubber, isoprene rubber, and halogenated butyl rubber. And some uses of the tires are for different types of vehicles. In this sample, we found styrene and bunch of other compounds. We found butene, so we call it styrene butadiene rubber. It also contained isoprene and limonene, which was indicative of natural rubber. So it will be SBr NR rubber in the sample. In order to confirm our analysis, I found a tire in my backyard. So I cut some pieces and run the GCMS analysis on the pyrolysis using pyrolysis. And it has butene, it had isoprene, it contains styrene and other aromatic compounds like we saw before. So we call it styrene butadiene plus natural rubber. I could not find the limonene peak in this particular pyrogram. We look at some new challenges in the pyrolysis GCMS. One of the frequently asked questions I get is what is the limit of quantitation and what is the detection limit, and how low can we go in the analysis? So the answer is very simple. If you can see it, you can do it. Dr. Keith Cooper, Dr. Beth Rabbit, and Dr. Brian Buckley from Rutgers University provided the samples of small microplastic samples from the Raritan and Passaic rivers in New Jersey. So as you can see on the left-hand side, there are tiny pink fragments. We found evenly spaced triplets, which is indicative of polyethylene. This is another green fragment. We found it has a marker peak of polypropylene. This additional small tiny pink fragment, it had a signature of polypropylene again, polyethylene, I'm sorry. And this dark blue fragment has signature of polyethylene. So the next question is, what if this detection limit of a fraction of a millimeter is not good enough? And what if we need to go even lower? So this question comes into picture when we work on small animals like oysters or other shellfish. For the mass spectrometry, size is important, and if there is not enough sample, it is hard to do the analysis. So here are some suggestions on how to improve the detection limits. We can increase the mass spectrometer auto-tune voltage. We can experiment with the GC injector port split ratios. If accessible, we can switch the analysis from a single quadruple to triple quadruple mass spectrometer or even to a high resolution mass spectrometer. For target polymer analysis and target additive analysis, switch from full scan mass spectrometry to select ion monitoring mass spectrometry. If nothing like this works, then you can switch from to FTIR or Raman spectroscopy as a potential exploratory tools. Here is an example of polyethylene, which is in a published paper by Thushari. It shows the Raman spectrum on the top of the fiber from the rock oyster, and the bottom shows the Raman spectrum from the library. It shows that this technique has a great potential, but as you can see, the spectrum is not very clean. It may have some fluorescence issues. So there is a need for improvement in the quality of the spectrum. Here are the students who contributed to the analysis: Darius Sanford, Nigel Lascelles, Dante Freeman, Jarrod Snook, Kelly Harmon, Julian Venable, Jessica Hemmelgren, and we really appreciate the students' contribution. So thank you for your attention. If there are any questions, I'll be happy to answer them.
Thank you so much, Ashok, for your very interesting presentation. And I realized that pyrolysis GCMAS is a very powerful tool for analyzing plastic samples. And lots of questions have been putting up some on slide. And oh yeah, so we go through one by one. So we'd like to go first for the most liked ones. The first question is from sorry for my pronunciation, the Baron. Thanks for the presentation. I have two questions. First, if I have a mixture of different plastics, will the GC mass give all signature at the same time?
Is that the only question, or you have a
Yeah,
second part of the question?
The question. The second question is, how do we quantify the signals we receive?
Okay, so the first question is, if you have two different plastics, can we see two different signatures? And the answer is yes. You can, as I showed you in a number of slides, you can analyze a polymer, a copolymer and the additive in the same sample run. So it is not a problem at all. And the second question is, how do you quantify? So in order to quantify, we need to develop a calibration curve, and then you can do the quantification based on the signals you get from the unknown sample. So suppose if you have polyethylene, you want to develop a 5-point calibration curve, and on that curve, you see where your sample falls, and that's how you will determine the quantification of that sample.
Okay, thank you. And I think some of the questions you have already answered in some part, but if you may want to add more explanation, so I'm going to ask a question. So the second question is from Barbara. What is the minimum size or mass of microplastic which that is needed to get a good signal with pyrolysis GCMS?
Sure. Yeah, that I kind of covered that. If you, it all depends on the instrument you're using and the sensitivity of the instrument. But what for a common single quadruple mass spectrometer, you can go a fraction of a millimeter without any problem. If you use a more powerful mass spectrometer, you can go to much lower detection limit. Like I showed in the triple quadruple, if you use triple quadruple mass spectrometer, or if you use a high resolution mass spectrometer, the interferences are eliminated and you improve the signal strength. So you can improve the detection limits.
Right, thank you. So the next question is from Corey. Thank you, Ashok. How does analysis of pyrolyzed plastic impact the integrity of the rest of the instrument? Does the inlet or column gunk up faster?
Right. The pyrolysis happens offline. So the pyrolysis does not happen in the mass spectrometer or the gas chromatogram. So the pyrolysis occurs in the quartz tube, and the degradation products, they are transferred in the gas phase to the gas chromatographic column. So it does not affect the cleanliness of the GCMS at all. And the quartz tubes we use, they are disposable. So we use a fresh quartz tube for each sample.
Mhm. All right. Right. So next question from Robert. Happy New Year. What types of halogenated polymer pyrolysis compounds do you recover from PVC or brominated plastics?
We actually don't recover any compounds. They are when PVC is pyrolyzed, as I showed in the slide, it eliminates the chlorine molecule and hydrogen molecule to form hydrochloric acid. And the side chains, and the double bonds rearrange, they cyclize, and they form aromatic compounds. So those are the kind of products we see, which are like totally unsurprising when we first started running PVC analysis. So we were not expecting that kind of um, spectrum um, character from the pyrolysis of PVC.
All right. And also from the same uh, questioners, are results from microwave assisted pyrolysis experiments comparable with pyrolysis GCMS?
I'm not familiar with the microwave assisted pyrolysis, so I should not say anything wrong. There are different types of pyrolysis instruments available on the market. So I gave you example of what I use, but the basic um, principle of pyrolysis remains the same. They just have the different types of parts. Some use a chamber to heat the samples. We use the quartz tube to heat the sample. So it depends on the manufacturer who makes that instrument. There are little variations of each instrument.
Right. Okay. So next question is from Timothy. Are the samples you received from community marine debris cleanups or field campaigns?
Yes, we did. We did. The samples I showed you from New Jersey, they were some from the beach cleanups. So yes, thank you for that question. Yes, we work with so many different types of colleagues that if we had to go do this work ourselves, it will be impossible for us to go to Alaska, to Hawaii, to Mexico, to various community efforts. So we depend largely on our colleagues to collect the samples and send it to us for analysis. And they are happy that we do this because they do not have the capability for the analysis, and we don't have the capability to go in the field. So it works best for the both. That's the part of the collaboration. It's beneficial to both parties.
Right. Also, yeah. Also, the also question from Timothy. Do you get most of your samples from? Oh, it's the same question. Sorry about that. Uh, Next question from Donia. Hello. Do you have a library uh, built built it built in to identify the polymer components?
Yes, that's it. It takes it's a painstaking process in the beginning. It takes we spent about 4 to 6 months in the beginning of our project to understand what pyrolysis is and how the spectra look like, how to identify. So yes, we have developed our own library of different polymers because those libraries can change from instrument to instrument.
Okay, thank you. Ashok, can you stop the sharing the slide now so that we can figure our faces? Yeah, thanks. Great. Okay, so next question again from Barbara. Are there any tricky natural polymers that have PUR fingerprints that are difficult to distinguish from synthetic polymers?
Uh, no, because that is the beauty of mass spectrometry. They're they are specific to a given polymer and depends. It does not vary whether you have natural polymer or synthetic polymer because each polymers it disintegrates in a unique fashion when they are subjected to heat in the pyrolysis unit. So we, as I showed you in one of the fabric uh, slide, there was a cellulose, there was a cotton, and there was a polyethylene terephthalate in the same sample. So we have a synthetic fiber and a natural fiber in the same sample, and we had no problem. So it it is based on the fragments of the polymer which we are analyzing. So it is we have not seen any issues with the identification regardless of the origin of the polymer.
Hmm, thank you. Oh, here's another question from Anonymous. Question is, how important is it to isolate the plastic from the environmental samples? For example, analyzing an entire leaf looking for microplastic or fraction of sediment.
Fraction of sediment.
Here, the question is, how how important is it to isolate the plastic from the environmental samples?
Oh yes, yes, yes. It is you have to the capacity of the quartz tube is very small, so you cannot put the entire sediment inside there. It has to be a certain size. So it is important that the plastic is extracted from the sediment and then it is placed in the quartz tube for analysis.
Hmm, right. Here's a question from Curry again. You explained how to quantify different polymers, but mostly showed quantitative results. Have you had much success with quantification?
We don't get into quantification at this time because we are right now developing methods for identifying successfully different types of polymers. Um, I always think that if you want to quantify a plastic, we can just weigh the plastic material on a balance, and then when we identify the plastic, you can determine the quantification based on that. It is not like a um, analysis of like a POP, like a PCB or PHS in the environment. They are like very small quantities in the fish, and we extract in the organic solvent, and then we do the analysis. So that is where quantitation becomes more important. But if you want to quantify microplastics, you could probably weigh the plastic, and then when you analyze what that plastic polymer is, you can say that this is the plastic that we have. That's the quantification I'm thinking about.
Hmm. Right, thank you. Next question is from Anonymous. I'm sorry if this has been answered. Is it possible to obtain the composition of those? Oh, so I repeat the question. Is it possible to obtain the composition, or does it just tell you the component present?
Um, Sorry, did not.
Is it possible to obtain the composition, or does it just tell you the components present? Component exist?
Yeah, you can analyze the composition, because when we have multiple polymers in a given plastic sample, you get the marker peaks for each polymer that can tell you how many components are there. In addition, you can also analyze the plastic additives and any other chemicals present there. So yes, it can be all done in the one sample run. I hope I analyze that, answer that questions to the satisfaction of the
Mhm.
reader.
Right, thank you. And guess that's all the questions. Any other question I haven't asked? Yeah, I think that's all the question we have now. And Ashok, do you have anything that you want to add or any comments?
Sure, I do have comments. The intent of my presentation is not to tell you what to use or what not to use. I want to remind the viewers that whatever they use, they should use it with an informed. They should make the decision based on the information, and not just use it because they have the instrument or the somebody else they know have the instrument. Because there can be pros and cons of each technique, and we should take that into consideration when we analyze a microplastic sample. It is very critical, because sometimes you see the papers where people say we could not analyze 20% of the samples. So question is, why did not they analyze the sample with other technique? And that is something I would like to emphasize in this.
Yeah, thank you. Oh, I uh, there is another question. I mean, it's from the previous one, saying I meant obtaining the composition quantitatively. I think it's a question from
Yeah,
the
yeah,
previous one.
no,
Yeah.
sure. That can be done as long as you can develop a calibration curve. Um, you take 5 point calibrations, like add say 1 milligram, 2 milligrams, 3 milligrams, 4 milligrams, and 5 milligrams of sample in different runs, and you develop a calibration curve. And when you analyze your own sample, you can get the response from that, and that's how you do the calibration, quantitation of what you have. If we have two components, then we develop the calibration curve for multiple components, and then see if what kind of composition we have, and what is the quantitation of those two compounds or three compounds for that matter.
Okay. Thank you. Oh, another question again. Oh, you have a lot of questions.
That's fine.
Questions from uh, Donia. Have you analyzed any microplastic in a small size range, like less than 10 micron or less?
The particles I showed you from Rutgers University collection, those are the smallest particles we analyze, and they are fraction of millimeter. That could be 200 micron size. So that is the lowest size we have analyzed.
Hmm.
But I know we have a colleague in Norway. They have purchased a high resolution mass spectrometer. So they probably have better capability of analyzing smaller particles than we can.
Okay, cool. Thank you. I think that's all the questions. Yeah, thanks so much, Ashok, for your very interesting presentation and wonderful answers. And I have a just announcement for everyone. The next Motion Plastic webinar will be February 9th. The topic will be the plastic survey from satellite. So stay tuned in the next month. So I'd like to thank Ashok again on behalf of the team of the Ocean Plastic Webinars for joining us today and for being available today and sharing your work. And I think, yeah, I think that's all for today. And thanks so much again, Ashok, and see you all in a month.
Thank
Thank
you
you
for the
again.
opportunity. Yes.
Bye bye.
Bye bye.